brick wall helped define part of the gravesite that we're talking about today. So here's a much later plat. German Wright owned all this property, and sometime in the 1740s, or late 1740s, early 1750s, he built a brick wall. That brick wall is described in newspaper advertisements by 1754. And that brick wall is just south of the burial site that I've indicated in that pink square. So the people that we're talking about tonight are buried on the west side of that wall. Did the presence of that wall have anything to do with choosing this location? Or was the brick wall put up to separate some kind of residents from this African-American cemetery? We don't know the sequence of events, but based on the archaeology and the DNA analysis that we're hearing about tonight, we're thinking, this is second half of the 18th century burials, and this brick wall is there by 1754. So what is the relationship between that brick wall and the burials? I don't know. German Wright tried to subdivide this property, failed, sold it back to George Anson, and George Anson later divided it up into some pieces. Most importantly, George Anson in 1761, the attorneys working for George Anson, sold this 3¼ acre property to William Ellis. That's where the bodies are, on Mr. Ellis's property. But Mr. Ellis acquired that property in 1761. Did he or his family or his heirs have anything to do with interring those people? We don't know. There's no paperwork documenting that. But that, again, is a plat showing you the location of the burial, that pink rectangle, and the brick wall. So those people are buried on the west side of that brick wall. Here's a 1798 plat, or a later plat showing you that in 1798, Mr. Ellis had died. His heirs subdivided that property. You can see the subdivisions of that property, and that star marks the approximate location of the graves. So this property got subdivided and people built over top of it. So there were no permanent markers acknowledging this as a burial place, and people built over it and forgot it. In 1967, the city of Charleston purchased this property because they were planning to build what they called a municipal auditorium. We know it as the Gilliard Center today. Here's Anson Street in this 1968 photograph, and that red star marks the approximate location of the burials. And you might look at that photo and say, what is that curious street? In the center of the screen, that's Wall Street. Wall Street mostly disappeared in the 1960s because of the Gilliard construction project. Wall Street, a little stub of it, still exists today. But Wall Street was so named, and it was created in the 18th century. Wall Street was just east of that brick wall. So where the photograph approximately is that brick wall, approximately where that red line is. So I think that we need to explore more in our study of the history of the site. We need to explore that brick wall and see if there's any correlation between building that brick wall and the interring of enslaved people of African descent on this site. It's an ongoing project, but I'm very pleased to have been working with Dr. O and this team for several years on this project. And I hope to have more information to share with you in the future. But tonight, we're going to hear some DNA analysis from the experts. I'll turn it over to Suzanne.
To the microphone. So I am Suzanne Abel. I'm the forensic pathologist. I'm a consultant with the Charleston County Coroner's Office. I'm a consultant in South Carolina. I'm also an adjunct professor of anthropology here at the college. I'm here to sort of follow up a little bit on what Eric and Dick have already presented. Give you a little further details on the skeletal and dental analysis that we conducted on the remains from Anson Street. So, I'm not used to the screen being behind me. What did I do?
You need to get to the right.
Sorry, guys.
To the right, to the left is forward.
Look to the right. Okay, there we go. So some of the skeletons were actually well preserved, able to utilize information how they lived? What was their lifestyle like? What was their health status? Because we're using skeleton to look at these things.
Can I talk a little louder? Okay.
I just want to say, is it even on? Can you hear me in the back?
Barely can hear you. The audio is not working back.
Hey.
Just gotta talk right into the mic.
We started feedback.
Talk right into it.
Testing.
Can you turn it like that? There you go.
Okay.
Oh,
Okay, here I am. Okay. So I want to say that most of the bones that you're going to see in these slides are not from individuals at the Anson Street burial ground. There are 2 slides that include people from the location. I'll let you know by giving you their burial number. Okay. So as anthropologists, when we're looking at the skeleton, sort of the interesting thing to me is how can we sort of recreate their lifestyle? Okay. So this is behind me one example of what we're looking at. So what we see here. The 2 bones on the left, that's the humerus, okay, that's the upper arm bone. And an arrow is pointing to what's called a deltoid tuberosity. Okay, this is a site where muscles attach on the arm. Okay, so 2 different individuals, right, 2 different upper arm bones. We can see the individual on the left, that deltoid tuberosity is much more built up than the individual on the right. Okay, guess what this means? Okay, this person on the left has much bigger muscles. Okay, the bone is going to reflect muscle size. The bigger the muscle attachment, the bigger the muscle that was on there. Okay, so we tended to see a lot of this with the Anson Street individuals. Well-built bodies, well-built bones, okay? And it wasn't just the humerus, okay? We saw it all over the skeleton. Even down to the little phalanges, the little tiny bones in the fingers, that we could see evidence for strong grip. People using their upper arms a lot. Okay. We also saw a few cases of what we can sort of generically call arthritis, degenerative joint disease. Okay, arthritis comes about for several different reasons in the skeleton. It can come from, repetitive chronic use of a joint. It can come from as a result of traumatic injury. It can result from just old age. As we grow and as we mature, we tend to get arthritis. Okay. The arthritis that we were seeing on these individuals was actually sort of focused more on the younger folks. So what does that tell us about lifestyle? That it was actually the younger people. Okay, so it's a repetitive use of the joints of the body and not necessarily age-related. So they were using their bodies vigorously. Okay. We also saw the occasional bone fracture. And you can sort of see this one right here. This is one of the individuals from Anson Street. This is burial 6. Okay, so this is a little phalanx. Again, one of the little tiny finger bones up in here, and we can see this fracture, well healed. Bone fractures are not uncommon in historical samples, just as they're just as common in contemporary samples. Really beyond these sort of minor skeletal trauma, minor reactive bone, we didn't see any real evidence of any kind of systemic disease in these individuals. And it could be because of preservation. Some of the individuals were well preserved, others weren't. This is burial 16. Some of them, this is what it looked like. And sort of this is what I'm given to sort of come up with. So you can see the before and the after. Okay, so this is what it looks like when you put it on the exam table. This is what you've got to deal with. And everything's covered with dirt and sand. After you rinse it all off, you have to rinse it to see the structures underneath. This is what you're left with. Okay? And it's common, given the date of the burial grounds, this is what you expect. But if you notice, it looks like there's no information left here. It looks like everything is just blown apart. It's all broken. What can an anthropologist tell? You can tell a lot from all this. And if you notice in particular, look at the teeth. Look how well preserved those beautiful teeth are. This was common with the Anson Street folks. Wonderful dental structures. You can tell this preservation. Okay? Because the teeth don't remodel after they're formed, they can't heal themselves like bone. All right? The teeth are this wonderful permanent marker of a life. We can tell sex, age, ancestral affiliation, diet, health, lifestyle,
cultural factors, all from the teeth. So in a population like this, where there can be some damage, preservation issues, it's the teeth that we're going to look at that's going to give us the host of the biological information for this group. Okay. In some of the skeletons we saw what are called, it's a fancy word, linear enamel hypoplasias. Okay, and that's just a fancy word. We're looking at these sort of horizontal bands on the teeth. You guys can see that, these horizontal bands. It looks like it's something that's etched into the teeth. This is actually sort of interruption of the growth and development of that tooth. Okay, and this is a common thing that's used archaeologically. It was high frequency in this sample. And what the linear enamel hypoplasia is, a sort of dental defect. What it represents, again, is an interruption of the growth and development of that tooth when that person was young. Okay? Now, we can't specify what exactly caused the linear defects, but historically, they come from overcoming childhood illnesses. They come from episodic malnutrition, overcoming these things, some sort of non-specific metabolic stress. So again, just looking at the teeth, it's giving us a picture of lifestyle, of health status. All the adults, every single adult, to some degree, expressed what we call occlusal attrition. Fancy word for saying the teeth are just sort of worn down. Okay? The older folks were worn down to such a degree that it was just flat, right up against the teeth, up against the roots there. It's a common thing that you see archaeologically. And in older collections like this occlusal attrition, this wearing down of the teeth, tends to come from grit in the diet. Okay, if you're grinding things, there's sand in the diet that's left over. If you think about these people, they didn't have access to these highly processed foods like we have. Okay, their bread was not Wonder Bread that was manufactured in a factory, it was purified, there's no sand or grit in it. Their bread would have been much more coarse. Okay, through time, it just sort of sands the teeth down. Sort of a common thing. I have to look up a cheat sheet here. Okay. Dental health was very poor among these individuals. 70% had dental decay. 43% were missing teeth, presumably following severe decay and abscesses like up here. Over 40% of the adults had one or more abscessed teeth. If any of you have had abscesses, you know how painful that is. Okay. And so all this decay tells us about their diet, which was probably high in sticky carbohydrates, things like rice, things that are decay causing. So by looking at the teeth, we can tell a lot of information about diet and health practices. So oral hygiene, access to dental care is either limited or really non-existent in this group. So now for some of the interesting cultural practices that we saw. Some of the people had modified teeth. Sometimes they were intentionally modified, sometimes it was unintentionally. Does anyone know what caused that gap between the upper and lower teeth? Pipe smoking. Yeah. So back in the day, there were the kaolin pipes, right, Eric? They were sort of these real gritty clay-based pipe stems. And so this habitually clenching that pipe stem between the teeth would have worn down the teeth in this really sort of characteristic way that we see. That's very common. Okay. What's interesting is I think it was like 10% of the population exhibited this. If we had dental structures to look at, we actually saw this quite frequently. And we also saw it not just in males, but also in females. That's interesting. Two individuals showed intentional filing or chipping of the teeth. Okay. This is a young lady sort of modeling what it looks like in life. So what happens are the teeth are intentionally chipped out with rocks, or they're filed down in a sort of purposeful, ritual type of way to sort of produce these unique tooth structures, these forms. Here you can see some of the different kinds that people have reported in the literature coming out of Africa. So tooth filing practices are frequently seen in and among different tribes in Western Africa and Central Africa, among other places in the world. And it's really thought to be sort of a sign of ancestral group affiliation. It's also done for aesthetics. Like we here, Westerners, we think that perfect teeth in this unnaturally straight row that are sort of white as a toilet bowl, we tend to think this is the ideal dental state. Well, other people think aesthetically this is the ideal dental state. So cultural variation, people do all sorts of interesting things to their teeth globally. The presence of this intentional tooth filing is interesting because it really confirms what we saw in the stable isotope analysis, which is that some of these people likely originated in Africa. I am not the stable isotope person, but I will report on what she found. Okay, so by taking samples, we're able to conduct some chemical analyses. We're looking at strontium, oxygen, carbon, out of the bones and teeth, and sort of performing some chemical analyses on these things, which helped us answer some pretty interesting questions. Where were these people born? Where were they living as adults? These are things that stable isotopes can help us address. Okay. So from these chemical analyses that we did, 3 distinct groups are identified. Okay, so those from Charleston, or at least born in South Carolina. Another cluster of people who recently arrived in South Carolina. And the last one, those who are more long-term residents of the state. So interesting with South Carolina, or what stable isotopes can do, and also a great demonstration of skeletal, dental, and chemical analyses all working together, like what we're going to see with the DNA soon. Okay. So lastly, some of you may be wondering about how these people may have died. There's really no skeletal evidence for anything obviously traumatic, and that could be because of preservation issues. There's all sorts of things that you can think about. There were some likely causes of death of people living during this time. Okay, sepsis, viral issues, natural causes, heart disease, and it could also be traumatic things. You have to remember that these people, as far as I know historically, are living in pre-antibiotic days. Okay, so how many of us would live as long as we have without access to antibiotics? So just a minor thing like a tooth abscess could be a lethal issue if it's not under control. Really all that. Thank you guys. Thank you for letting me be a part of this really important chapter in Charleston's history. Thanks to Brockington, the city officials who've been nothing but gracious about everything. If anyone has any questions, I will gladly address them afterwards.
So archaeologists have a way of talking about bones and teeth and stuff like it's sexy. You know, so this is what we do anthropologists. We do research and we like to share our research, and it's not often. It's not often at all that we get to have the public to share our research with. So I want to thank y'all for being here for us to share our work. So on that note, I want to thank some people before people might need to get up and leave. Firstly, I want to thank the National Geographic Society. For those who don't know, National Geographic is sponsoring some of the research that Gullah Society is doing on this DNA. And tonight we have a National Geographic writer here to report on this evening's activity. We have Adeyemi, who is a National Geographic explorer, scholar, as a result of his participation in this project. And I am also a National Geographic explorer scholar through this project. And so I thank National Geographic, I thank the Coastal Carolina Community Foundation, of course I thank the City of Charleston, Eric Poplin and Brockington Associate, Butler, and the library, Suzanne, thank you very much. Tad and Raquel, thank you. Adeyemi, of course, thank you. Heather, thank you. Gil, Joe, Gil, thank you. And if I miss anyone, please excuse me. And of course, oh, yeah. Well, I was just reminded that I needed to thank Joanna Gilmore, and I guess I should thank myself too, right? No? If I'm thanking Joanna Gilmore, I have to thank myself because, like, you know, for me and Joe, that's what she's known as in my circle, this has been a really a heartfelt and sometimes tireless effort. And maybe 1 year ago, on this very same project, we was doing what we call community conversations, and we had occasions when there was just 2 people in the room. We've had occasions when it was Joe and I and 3 people in the room. But we just kept on pressing. And there was one occasion where we had a caterer to come and cater the event. They catered and brought 30 meals, but we just had 3 people to eat those 30 meals. But we were kind, we fed a lot of people that day. So, again, thank you for your participation. And those departments at the college who contributed to this cause, we thank you very much. And if you look on your program, there's a list of people, departments that participated. And I'm going to, and I'll say this now, invite you to please pick up your program. And inside there's a questionnaire that says, share your thoughts. Please, we have pencils in the back, and this supports our work because your thoughts will determine, or partly determine, what the memorialization of those ancestors will look like. So please, share your thoughts. And yeah, so please, take a few minutes to do that because this is critical to our work. All right? So I'd like to invite Dr. Theodore Schurr and Adeyemi Oduwole to come up and share with us their research findings.
Hello, everyone. I'm Theodore Schurr from the University of Pennsylvania, the Department of Anthropology, with Adeyemi Oduwole from the College of Charleston. And we're going to be giving the first half of the genetics part of this data and explanation, followed by one on the focus on ancient DNA work with Anson Street burials that will be explained and outlined by Raquel Fleskes, who works with me at the University of Pennsylvania. So we're going to first give a talk about the ancient DNA work and try to show how it connects to both the modern DNA work and show how it connects to ancient DNA work. What I'd also like to say is, following the comments by the archaeologists Eric and Suzanne, is that part of what we're all trying to do with the biological materials from the graves is to basically bring back to life the history of these persons, the biohistory of these persons. And different parts of the body are chemicals or DNA that is able to be extracted from these remains tells different parts of the story. So it's actually quite crucial that we're working together to be able to do this, because we may actually see some things from the analysis of teeth which we don't necessarily see from the DNA data. The DNA data may tell us things we want to relook at, re-examine from from the archaeological or the osteological, the bone, or the dental part of the story. And of course, DNA is not going to say anything about pipe smoking, it won't show you anything about the teeth, but it may say something about how far and wide your ancestors have come and who you might have married or had ancestors in the recent or even deeper past. So I want to make sure the DNA evidence is part of the story, but it actually, we need all the other archaeological, historical context to be able to properly interpret what we're doing. So for this presentation, this part of the presentation, we wanted to give, since he played an essential role in this DNA analysis, working with Raquel and I, carrying out the DNA analysis. And actually, before we even start, I'd like you to give him a hand, because he stepped into a big project. We stepped into this very big project to do work with relatively untested in the laboratory. And to learn very quickly, excel in the analysis that he carried out, and is going to make a big contribution to what we're able to finally say about the genetic diversity of members of the community here of Charleston, but also how they in some ways are connected biologically, genetically, historically, ancestrally to the individuals who are buried at Anson Street. So I'm going to hand the microphone to him, and then he's going to carry forward the first part of this presentation. Then I'll come back and give some broader context for the DNA evidence we have so far.
Can everyone hear me? Yeah. All right, so as Dr. Schurr said, this is such a big project, but my portion of the project just focuses on mitochondrial DNA analysis of individuals living in Charleston. And as he said, all four parts of this project, the Anson Street burials, the genome chip analysis, mitochondrial DNA analysis, and the community engagement portion, all focus on African American ancestry. As most of you know, Charleston's important. Being that during the transatlantic slave trade, about 50% of Africans who were dislocated from Africa came through Charleston. And as Mayor Techenberg said earlier, the city of Charleston has apologized for their role in slavery. So I've included that here in the slide. So you can see it's really important that individuals are building on with that in order to find out more about themselves and to understand what's really going on here. Mitochondrial DNA. As most of you probably don't know, mitochondrial DNA is inherited through your mother, and that's why it's important in tracking ancestry. Unlike nuclear DNA, the DNA that you normally hear about, that's taken from both your mother and father, so it's kind of like a mixture of the two. But unlike that, mitochondrial DNA is simply passed on from your mother to each offspring. So your mitochondrial DNA is likely or almost exactly the same as your great-grandmother or your great-grandmother. And this is important because you can use this as a tool to look at your ancestry. So the first step in doing this was we created our hypothesis, and that was by assessing the genetic diversity of African American people living in Charleston will provide you insights to the history of Charleston and the African diaspora and how Charleston played a role in it. And our first step in doing this was hosting DNA Day in 2018. And in doing this, what we did was individuals who wanted to participate in our study met up with us, and we were able to take bugle swabs from them. We actually took 2 bugle swabs, one for my mitochondrial DNA analysis and another for genome chip analysis. So these are the steps I used in the lab at the University of Pennsylvania in order to carry out the analysis. The first step was extraction. This is just a picture of the lab setup while we're extracting the DNA, the bugle swabs. The next step is PCR amplification, and we did this through gel electrophoresis. And most of you probably don't know what that is, but it's where we break down the DNA with different reagents and chemicals just in order so we can visualize it. The next step was cycle sequencing, and the last step was data analysis. And this was probably the most tedious step for me because it's really just sitting at a computer all day and looking at mutations on a computer and just analyzing and determining what mutation it was. As I said, all the lab work was done at the University of Pennsylvania in Philadelphia. And this is a photo of me and Raquel, who's here. She'll tell you about the modern DNA lab. said, this was probably the most tedious step, which was sequence characterization. So what we determined here was mutations found in the hypervariable segment region of the mitochondrial DNA. The first and second part of that, we were able to determine different mutations, and a string of those mutations determines an individual's haplotype. And from a haplotype, you can determine what haplogroup you're from. And through a haplogroup, that tells you what your lineage is. Through that, you can almost tell like where you're from. And as Dr. Schurr said, this is a haplogroup map, and it just shows you like different areas where like you'll find L haplogroups, so different areas where you'll find M haplogroups. And as we were hypothesizing, the majority of the individuals in our studies will fall under the L haplogroup, which are found normally in Africa. And these are the results from the modern DNA that I analyzed in Philadelphia, or from individuals in our study in Philadelphia. As we expected, a lot of our individuals were from the L haplogroups. So these right here, all of these were individuals who fell under L haplogroups, L1, 2, and 3. And then we had a couple alloirs right here, which were U6 and F3B1 haplogroups, which were also found in Southeast Asia and Southwest Europe, I believe. And this is just a bigger breakdown of the individual haplogroups, like the L1, L2, and L3 haplogroups, which are found in Africa. This map right here displays haplogroup diversity in Africa, and it just displays what we were looking for, most likely going to compare our data to once we discovered what haplogroups individuals in our study were.
Thank you for that. So just to backtrack briefly about a couple of things. So when we're tracing our maternal ancestry, part of the assumption is that we all have common roots, and those common roots are actually in Africa. And so the ancestral portion, the root of this tree for all humankind actually is in Africa. And then people dispersed within Africa and outside of Africa, their mitochondrial DNAs continue to evolve and change through a process of mutation, giving rise to all this rainbow set of colors and branches that represent the diversity of the rest of the world. The mutations that Adeyemi mentioned are actually markers which tell us which lineages that the DNAs belong to and where those mitochondrial DNAs fit within this particular tree. So basically, it's genetic genealogy. That's essentially what we're trying to do. And the markers that he described, and all those letters and numbers, are ways for us to designate what lineage that a person has based on the mutational character, the markers that their DNA has. So what I'm going to do, so this is an important point because the patterns of diversity across Africa are not uniform. Africa is an extraordinarily, as I'm sure you're aware, extraordinarily diverse place, linguistically, culturally, and certainly genetically. And we're getting more and more information about this as we study more and more populations from Africa or African descendant communities in different parts of the Americas and other parts of the world. So what this diagram is showing, and this is published in 2002, but it remains true today, is that Africa basically because we've been in Africa, modern humans arose in Africa 200,000 or 300,000 years ago, and basically spent a couple 100,000 years there before moving outside of Africa for over the next 75 to 80,000 years. So populations that spread all around the continental area there and diversified in different ways, leading to different patterns of mitochondrial diversity, which are sort of regionally based. And the fact that there are concentrations here of certain kinds of L2 types, that branch, may suggest that perhaps some of these mitochondrial DNA might trace back to that general area. Likewise for L1 and L2 and other L3 that he also was mentioning here. So this is a key to understanding where we might find possible ancestral roots tied to certain geographic areas, even if somewhat imprecise to begin with. Now part of the reason why this is complicated is because people have been in motion in Africa for a long, long time. And that's true, even now, migrations up and down throughout the continent, migrations out of the continent to Europe, back and forth across the Horn of Africa, and so forth. And this long process of movement is actually mixed, even though these lineages are have a sort of, we can see them in a tree-like form, the genealogical form, where they get sorted out and dispersed across Africa. It actually is quite wide, based on human movements, sometimes really substantial movements like Bantu expansion, Bantu-speaking groups, 5,000–6,000 years ago, maybe a little bit later than that, spreading all across this region here and transforming that area genetically. So our task is, the task of finding roots to Africa among persons living today is complicated by the complex history of Africa and African peoples themselves. So it is a bit of a detective story and an effort in this kind of way. I should also mention just a little bit of, if I can just quote a little humor here with Adeyemi. I think he had sort of this notion of CSI. You come in, throw something in the machine, you crank out a sequence, and then you've got an answer. And in fact, I think what he's come to appreciate is this work is, I started working on these kinds of data for 30 years. And there's still new things found every day with different publications. It took a long time even to get a basic understanding of African diversity based on the type of methods that we have now. Now we can sequence a genome basically in an hour. Your entire genome can be sequenced in an hour. But you're going to spend another 10 or 15 hours or longer figuring out what it means, or even more than that, and using computers and so forth to do that. So it's a computational work, which is the tricky part, in which he gained a lot of expertise and should be given credit for diving in and tackling this with not really all that much familiarity. It takes time to learn how to interpret the evidence that you have. So this is an ongoing story. I should also mention parenthetically that one of the reasons why we've not known so much about African history is despite the fact we know that our roots lie in Africa, until some years ago, 5–10 years ago, there wasn't a lot of studies that actually conducted that involved African populations. It was assumed that we knew what was there, or there was a blind spot, Europeans analyzing Europeans, as is typical also by medical work. And it turns out we don't have all the answers by studying Europeans. So the history that emerges from this is far more complex than people understood. Now we can link it to archaeological, cultural, linguistic histories in ways which are much more nuanced and sophisticated. And trying to find roots now is made somewhat easier by the fact that we now have hundreds and hundreds of populations of African descent, contemporary Africans, to help us find out what kind of connections there are between peoples living of African descent here and African populations living today. The ancient DNA data, which Raquel Fleskes will talk to you in a moment, also helps looking at the deeper past, whether there's a continuity from past to present or whether there's been change and what that actually means. So that's the part of the story which is really coming forward, is going to change our view about African history, but also about African American history in this part of the world, also in the Caribbean and in South America. So with that background, let me just go in a little bit here. And of course, this is a map that many of you are familiar with. But the idea, of course, is that while we know that people were forcibly enslaved and brought to the Americas here, sometimes from very specific sorts of places, what we're trying to do in essence is sort of reverse engineer these past connections where people have come from using genetic tools and using other evidence which tells us about this process of enslavement and exportation into the world. Now, I wanted to show this map because this is also helps to emphasize the point about regional diversity. So all this is doing here is giving you a color coding for these female lineages. This is a maternal ancestry. So the lineages that we see most commonly among the members who participated so far are these green or orangish colors that are here. And you'll see that some have very strong frequencies here. Others you see that they're less frequent as you go around Africa. In part because North Africa has been connected to the Mediterranean here, East Africa connected to the Arabian Peninsula and to West Asia. This area here also has been somewhat distinct, as in South Africa among the Khoisan speakers. And Madagascar actually was first settled by Polynesians before mixing with African communities some couple thousand years ago. So each of these areas of Africa, which is always shown to be smaller on most world maps than not, actually shows an extraordinary diversity. But the fact that we're finding these lineages here, these maternal lineages, primarily in these regions, suggests that this is the likely areas from which individuals who pass these mitochondrial DNAs down to you all actually may have lived at some point in the past. Now this lineage here, which Adeyemi mentioned, is actually one that is interesting enough, is very common among North African populations, including Berber populations. And this probably had actually spread through Sahel, through the sub-Saharan regions, through various contract and trade routes and things of this sort. So it's a relatively unique one in the sense of being North African, but it is someone who participated actually had this particular maternal lineage. And so also what I wanted to mention is that in terms of tracing deep ancestry and connections, part of the complication is that this is an ethnolinguistic map of represents the sort of 4 or 5 major language groups. And then these little names here are all different ethnic diversity that exists that we can map now. And part of the problem, while this shows this great diversity, lots of people have been moving around for some time. So pinpointing the exact ancestor to some degree is going to be a little more complicated because of this complex, this ethno-linguistic history of Africa itself. We also found a couple of what we call F3Bs. So this is a tree showing the genealogy from the ancestor of the F3 branches here. The interesting part of this that we found actually is that F3s tend to fall within, broadly speaking, within this area of Asia. So this is a little bit of a surprise, but these are in fact the data that we found, and we're still trying to determine what these data mean. So with that mitochondrial work then, we're happy to answer any questions for you afterwards. So what we could do with mitochondria actually shows us a maternal lineage, but it's not the whole picture. We ideally want to know what all our DNA markers in our genome tell us about ancestry. So this genotype analysis, that's the name of this particular tool. It actually surveys markers across all your chromosomes to find out what ancestry you might have from various parts of the world. These are markers which are relatively high frequency in various parts of the world, and then you're basically screening your DNA. You determine which of those markers you have to have. And therefore, which part of your ancestry might come from Europe, might come from Africa, the Middle East, wherever this might be. And so that's the basis of this particular tool, and we're working. I called up the lab today, and the data are still not ready, but we hope they'll be ready by about a month's time. But basically what this is going to show you is that wherever your ancestors trace their roots to, we're going to actually be able to see some evidence of these contributions to your own overall DNA. So why do we do this? Well, what we can do with this is use this is sort of what's called an admixture plot. So and this is a little bit technical, but I think what you get the idea is that we're saying how many markers. Let's say these are a variety of reference populations here. So we work with a number of people of African descent, a number of Native Americans, a number of East Asians, Europeans, and South Asians, and then ask for all of us who are surveying our DNA, which of these 5 major sets of markers does our DNA have? And then what we can then do, although you may not be able to see it very closely, these are some other populations from the Caribbean, which show mixtures of these types of backgrounds because of their own complicated ancestry that includes Native, African, European, and even South Asian. So we wind up having sort of a series of, you can see these persons have a mixture of blue of Native types. Some individuals have green, which represent European mixture. Some have African ancestry, depending on where they're from in the Americas. So this is going to tell us that we can categorize this and say that we have so much European, Asian, and so forth mixture that tells us about this biohistory. We can also possibly even map out where on the chromosomes we get these different DNAs from different parts of the world. So this is a person with European ancestry, but this person has ancestry from different parts of Europe and different parts of the Middle East and so forth, and this is called chromosome mapping. So this is getting into the weeds here, but it tells you the sort of level of resolution we can get with the DNA data to tell us the story that certainly with living persons. And what we hope to be able to do is something similar with ancient DNA data in the very near future. So what does this do? Well, we can inform ancestry for persons living here today, and also individually. This may help in terms of each individual understanding where his or her ancestry. And the future directions will be that we continue to do more work with other individuals from Charleston to expand the data we have to get more representative diversity from the community, and also ultimately be able to compare the data from living persons and participants with the data from the ancient DNA sample that is from ancestry variants. So before turning over the microphone to Raquel, I'd like to thank, I also have some thank yous, including the Raquel Society and others who funded this work, all the institutions who are involved, and also especially the City of Charleston for their support of this project. So I'm going to turn this over to Raquel, and thank you for your attention to this presentation.
Before Raquel, I just wanted to say that we have food that we ordered for y'all to eat, because I understand that some people came here directly from work. So if you would just, Raquel is going to come up and speak, and if you would just allow us 5 minutes for our back to bring the food in, I'm sure you'll enjoy it. So please just be patient with us. Thank you.
Hi, everybody. My name is Raquel Fleskes. I'm from University of Pennsylvania. I am an attached course student. Let me just get my notes all in order. I'm really excited to be here today to talk to you guys about the DNA analysis of the Anson Street individuals. Now, we don't actually have data right now, but we have good quality data, and I'm going to talk to you guys kind of about the process and the project goals and the research we've been undertaking over the past 2 months. So, as you heard from Eric and other researchers affiliated with this project, when the Anson Street burials were uncovered back in 2013 on the corner of George and Anson Street, they recovered 36 individuals put into rows of 4. Now, there were some archaeological artifacts associated, as we learned before, but importantly, none of the artifacts had any individually identifying information. So there are no headstones, no grave markers, no buttons with names. And as we also heard from the documentary evidence, there's been no individually identifying documents found at the site. The previous analysis done by Dr. Abel, as we heard, did osteological assessments of the burials. So we find a wide range of ages, going from juvenile, so here you can see the teeth coming up, so this is a young person, to an adult, look at this nice set of teeth, and then also older adults. Ancestry estimations indicated that 33 of the 36 individuals were likely of African descent. You can tell this by looking at craniofacial morphology of the bones. In addition, we also see that there's a wide, both relatively equal ratio of males and females at the site. And yet, with all this information, we still have the questions of how are these individuals related to each other? Do we see that between these 4 rows of individual interments, do we have any kind of kinship connections? Are there family relationships? Unfortunately, osteological data and archaeological data cannot tell this information. In addition, although we're able to tell broadly that these persons may be of African descent, where potentially in Africa do these persons have population affinity with? So are they coming from parts of West Africa, East Africa, maybe Central or South Africa? In addition, in previous looking at the teeth of the bone, you see here these teeth have kind of these indents right there. This is called shovel-shaped incisors. This is a characteristic Native American trait also found in Eastern European and Central European populations. So potentially we may be able to see on a finer grain level any sort of admixture or different ancestries in these persons. So this is why we were brought up to the project. So I've divided our project goals into twofold, one being scientific and the other one being community, but both of these kind of, I think, go hand in hand and largely build off each other. So what do we want to find out with the Anson Street individuals by doing DNA analysis? One, we want to characterize kinship. So we want to be able to tell, are there persons related to each other at this burial ground? Maybe is this a family burial ground, or perhaps is this an annex burial ground? This is the type of information we'll be able to tell. In addition, we want to characterize ancestry. So we want to figure out where might people have population ancestral affiliations with in the Anson Street burials at a very relatively specific geographical level. In addition, as Adeyemi were talking about previously, we want to be able to see how is the population genetic diversity of the Anson